Hi, my name's Angus, and I'm here to tell you about some exciting new research that I'm doing at the University of Newcastle through a scholarship provided by the American Society of Plant Biologists. In this research, I'm developing a new technique to develop the angle of microscopic structures within plant cell walls using polarized light. Now you might think after that, using light to measure the angle of something, that doesn't seem to make sense. Well, to understand how this works, we need to understand what these structures are that we're interested in measuring. So a plant cell, unlike an animal cell, is surrounded by a thick cell wall. And it's this cell wall that gives plants their strength, rigidity, and durability. The molecule within the cell wall that gives it its strength is called cellulose. Cellulose is made up of long chains of glucose sugars, and these chains bind together in the cell wall to form very strong fibres. And it's the angle of these fibres in the plant cell wall that we're interested in measuring, because as we'll see a little bit later on, it is very important. The technique that we're using to do this uses polarized light and fluorescence. And to understand how this works, you need to know what polarized light actually is. So you can think of the waves of energy in a light ray as vibrating on two perpendicular axes. Polarized light is simply light where one of these axes is removed, leaving light vibrating on only a single plane. This is achieved using a Polaroid or polarizing filter, like is commonly found in sunglasses such as these. So the Polaroid filter that makes up the lens of these glasses is made up of long chains of molecules that are all aligned parallel to one another in a given direction. So that when light hits the lens, the light that's vibrating on the same plane as the molecules will be absorbed. The light that's perpendicular to the molecules will pass straight through, creating polarized light. Now, the long fibers of cellulose within plant cell walls are usually aligned parallel to one another, just like in a Polaroid filter. So what we can do is stain these fibers with a dye that produces fluorescence when it absorbs light, a fluorescent dye. So if we stain cellulose fibers within plant cell wall with this fluorescent dye and image it under a microscope using polarized light, the fibers will only absorb the light and produce fluorescence if the polarized light is the same angle as the fibers. Just like in a Polaroid filter, if the light's perpendicular to the fibers, it will pass straight through and no fluorescence will be produced. So what we can do is take a piece of cell wall and stain it with this fluorescent dye and image it using polarized light at a number of different angles. And the angle of the polarized light that produces the most fluorescence will correspond to the angle of the fibers within the wall. So that's how the technique works. But why is this important? Why are we interested in these structures? Well, it's the angle of these fibers within the plant cell walls that determine how cells expand, how they grow. And it also determines the structural and physical properties of the tissue that the cells make up. An example of this is in cotton cells. The long cotton cells that make up cotton fibre have very complex cell walls, and it's the angle of the micro-fibrils or the cellulose fibres within these cell walls that determine the physical properties of the resulting cotton fibre. So if we can develop this technique effectively, it will have great practical use for plant research, understanding how cells grow and expand, and for fibre-based industries such as cotton to better understand what the structural properties of a cell wall are that make up a good plant fibre. So there you go. You've learned what makes up a plant cell wall, what polarized light is, and how it can be used to measure the angle of these microscopic structures within plant cell walls. If you want to find out more about the American Society of Plant Biologists and the summer undergraduate research program that made this research possible, you can find links in the description. Thanks for watching. I hope you enjoyed that, and most of all, I hope you learned something new from it.